Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I'm one of the founders of Striatech. I welcome you to another of our journal clubs where scientists present their project and data, highlighting the use and the application of our products. Striatech is a dynamic biotech company from Tübingen in Germany. We specialize in neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Today, it is my pleasure to welcome Kyoung Kim as our speaker. Her talk is entitled RIP1 Inhibition Protects Retinal Ganglion Cells in Preclinical Glaucoma Models. Kyoung Kim is currently working as a postdoctoral researcher at Hoffmann-La Roche in Basel in Switzerland. She has recently been awarded her Ph.D. from the École Polytechnique in Lausanne in Switzerland. Kyoung was the recipient of a Roche doctoral fellowship. This allowed her to perform her experimental Ph.D. work both at the labs of Roche in Basel and at Genentech in South San Francisco. Her Ph.D. focused on the interplay of different cell death pathways in retinal ganglion cells across diverse ocular disease models. One of these preclinical disease models is glaucoma. Glaucoma is one of the more common eye diseases, but still to this day, it cannot properly be treated. In her work, which was published earlier this year in the Nature journal Cell Death and Differentiation, Kyoung identified a promising new target for treating glaucoma, the protein kinase RIP1, and she will tell us all about this project today. Before you start, Kyoung, let me remind the audience that you will be able to ask questions throughout the event and also give a thumbs up to other questions. We will get to those questions at the end of Kyoung's presentation. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field to start typing your question. The Q&A panel on the right-hand side of the screen will stay available for a couple of days after the live event. So if you're currently not watching live, but a recording of an event, and if Q&A is still available, please make sure to enter your question and your name so that we can get back to you with the answer. We are very much looking forward to your questions. So Kyoung, please, you can go ahead and start and share your presentation. You can unmute your microphone, don't forget this. And I am very much looking forward to your presentation.
So hello everyone. So I would like to thank Striatech for inviting me to present here today. I am very happy to be part of this seminar series. So today I will be presenting our recent study that investigate the role of regulated inflammatory cell death in clinical glaucoma model. So before diving into the data, I really want to.
Kyoung, you still need to share your screen. I'm sorry to interrupt. Mm-hmm. It's not shared yet.
Yeah. Is it clear?
Yes.
Good.
Thank you.
Yes. So I will be presenting our study right now, but before diving, I really want to express my sincere appreciation for the individuals and teams who are really instrumental in making this work possible. So first, I would like to thank my PI, Domagoy and Peter, for their support throughout this complex cross-functional project, which also really involved in international relocation. And it was really amazing opportunity and their effort bringing this project to completion. Also, I'm very grateful to Joshua and Philip for sharing their expertise and engaging in valuable discussions that significantly contributed to the success of the project. Also, additionally, I want to extend special thanks to Tanya, Sebastian, Tom, and also Stella for their invaluable technical expertise and input. So before we really start with the data, let me briefly highlight that I hope you will take away from today's session. So first, we will reframe the glaucoma as a disease that goes beyond intraocular pressure, involving neuroinflammatory mechanism. Second, I will introduce RIP1 as a central regulator of necroptosis and inflammation in RGC degeneration, especially in preclinical glaucoma model. The third, I will examine how RIP1 inhibition protect RGC structurally and functionally in both injury-based and genetically disposed glaucoma model. And then we will explore how OPTN dysfunction, including the E50K mutation, sensitizes cells to RIP1 dependent cell death pathway. Finally, I will summarize how targeting RIP1 offers a unified and promising therapeutic approach across distinct glaucoma stress context. So let me begin by explaining the disease we are focusing on today. Glaucoma. So glaucoma is a group of progressive optic neuropathies. What they have in common as a hallmark is the degeneration of retinal ganglion cell and also their optic nerves. Especially RGC is a part of the central nervous system, meaning once they died, it has very limited regenerative capacity. So the prevalence is increasing rapidly. It's projected that by 2040, over 112 million people worldwide will be affected. Despite this high medical burden, there is no curative or preventive therapy available currently. And the only clinical intervention is to lower intraocular pressure, but IOP, intraocular pressure, does not fully explain the disease progression in many patients. So key characteristics include RGC loss from peripheral side and high IOP, but not all patient population shows this phenotype, and impaired blood flow, and altogether this feature leads to really irreversible vision loss in the end. So on the right side, you see the difference between normal optic nerve head and the one that affected by glaucoma. The cupping and thinning of the neural ring is quite apparent. So these imaging assessments are routinely assessed in clinical settings, but they only reflect damage after it has already occurred. And that is why understanding the upstream molecular drivers is very critical. So importantly, glaucoma is multifactorial, while family history and aging are established risk factors. Environment and molecular contributor are also being increasingly recognized. So the question is, what are the mechanisms driving RGC death beyond the IOP, and how we can target them? So traditionally, glaucoma has been understood as a disease driven by intraocular pressure, or IOP. So this IOP damages optic nerve head and leads to RGC death together with degeneration, so leading to the permanent vision loss throughout the time. But this IOP-associated glaucoma patient shows ocular pressure usually higher than 21 millimeter of mercury, and this is the major disease class in Western area. However, optic nerve damage and RGC loss can occur with normal range of pressure, usually 10 to 21 millimeter of mercury. So clinical observation of normal tension glaucoma is and progressive RGC loss, despite of pressure control, suggests there is additional mechanism play a role to induce RGC loss. So this type of glaucoma is a major disease class, especially in Asia, More than 76% of glaucoma patients diagnosed without clear IOP elevation. So even if they are different characteristic, they are common in clinical manifestation, RGC loss, together optic disc degeneration. So the key question is, why do RGCs continue to degenerate in the absence of or even despite control of IOP? So IOP is a part of the story, but clearly it is not the whole story. So understanding what drives RGC loss beyond the pressure is essential for developing new therapeutic strategy. So as I explained in previous slide, the glaucoma is more than an eye pressure problem. It is a complex chronic neurodegenerative condition with no current cure, largely because of we intervene too late in the disease process, or we target a just side consequence of the disease rather than the targeting onset. So this schematic illustration shows the disease progression from the healthy retina to the complete optic nerve head degeneration. In the early stage of the glaucoma, RGC begin to show sign of dysfunction. So these functional changes in RGC occur before any anatomical loss. So these can be detected by electrophysiology, even when patient notice any symptoms. So patient and often clinician miss this window. Another early changes is a decline of Basel perfusion even before substantial RGC loss occur in glaucoma patient. So this means the pathological cascade involves not only neuronal stress, but also accompanied with compromised blood flow, likely amplifying the RGC vulnerability. So only after a significant proportion of RGC have died, usually around more than 30%, we begin to detect the structure thinning and visual field deficit. And by then, this damage is really irreversible, as we cannot resuscitate loss of RGC. And this is why we must target the upstream mechanism before RGC die, before the structure is altered and before vision is significantly compromised. So if elevated pressure is not a single driver of glaucoma, what else contributes to the RGC degeneration? So, and also what molecular mechanism exists behind of the RGC death? So recent studies suggest that neuroinflammation is a key player, particularly through the microglial activation and cytokine signaling. At the molecular level, RIP1 emerges as a central node interesting for both cell death and inflammatory signaling. Through its scaffold and kinase function of RIP1, it can be both triggering necroptosis and also amplifying inflammatory pathways such as NF-kappa B. So this raises a question: if RIP1 is this is central, could this inhibition confer a broad protection against RGC loss across different models of injury, especially focusing on glaucoma? And what is that is what studies set out to answer throughout this storyline. And therefore, we hypothesize that RGC degeneration is not just due to mechanistic stress, such as intraocular pressure. It occurred together with both inflammation and cell death, so which is defined as an inflammatory cell death. So let's now look at directly to the human histological evidence. So in postmortem human glaucoma retinas, we observe a clear upregulation of inflammatory and cell death markers compared to non-glaucomatous control. So EBA1 positive microglia are more abundant and more deeply infiltrated into the inner retinal layers. This microglia infiltration, along with a high TNF-alpha level, and indicates a pro-inflammatory status. So RIP3 expression is also elevated in glaucomatous retinas together with TNF-alpha transcript. This supports the hypothesis that pro-inflammatory mechanisms, including cell death, are engaged and also necroptosis engaged. So also in parallel, Cleave's caspase 3A, which is a key marker of the cell death, is also upregulated in human glaucomatous retina. So in this slide, taken together, these findings demonstrate that the human glaucomatous retina is on inflamed status and undergoing regulated inflammatory cell death. So this supports our hypothesis to explore the RIP1 mediated pathway as a starting point. So to understand the necroptosis, we need to zoom in on its core molecular signaling mechanism. So the pathway begins with the TNF-alpha. Usually glial or microglia is a source of TNF-alpha. And then TNF-alpha binds it to the receptor called TNF receptor 1. Upon activation, TNF receptor 1 recruits TROD, which serves as a cytosolic binder for RIP1. And RIP1 is then modified through the multiple site ubiquitination by cellular inhibitory apoptosis proteins and forming what is known as a complex 1. So this complex activates NF-kappa B, promoting a pro-inflammatory as well as a pro-survival status. However, when deubiquitinating enzyme comes in, such as a CYLD, remove these ubiquitin chains. So the RIP1 transition away from the survival pathway and into the death signaling. So at this point, RIP1 can translocate it into the cytosol and then interact with FOD and caspase 8 and initiate apoptosis. This is referred as the complex 2A. So if caspase 8 is inhibited or absent, so RIP1 instead oligomerize with RIP3 to form the complex 2B. So this complex further phosphorylates and activates MLKL, which oligomerize at the cell membrane and causing membrane rupture, and the release of a damage-associated molecular pattern called GAMP, and this process results in necroptosis. So in summary, RIP1 function as a molecular switch that determines whether cell survival or undergoes apoptosis or engage into the necroptosis. So how might RIP1 mediated cell death be relevant in glaucoma? There is a mutation, is autosomal dominant mutation called OPTN E50K mutation, which is a known glaucoma causative genes. And several studies suggest that this mutation strongly binds to the TBK1, which is also known as a negative regulator of RIP1 dependent cell death, and it cannot be dissociated from it. So while the exact mechanism remain unclear, this dysregulation could shift the balance toward more RIP1 activation and increase the susceptibility to necroptosis in retinal ganglion cell. So now we have discussed how PTNE50K may dysregulate RIP1 signaling through TBK1 interaction. But how does this mean in real patient? So here I'm showing you the data from the research team from Moorfield Eye Hospital in London. The study with a glaucoma patient who harbors OPTN E50K mutation and tracked their progress of glaucoma. And this study shows that glaucoma patients with this mutation have a significantly thinner neuroretinal ring compared to those without the mutation. So both temporal and nasal region are affected, suggesting progressive structure degeneration. And also from systemic biology perspective, OPTN emerges as a highly confident disease-associated target for glaucoma, and it is centrally located within a glaucoma-relevant regulatory network. And finally, postmortem transcriptomic data shows that OPTN expression is significantly decreased in sporadic glaucoma cases. So it represents the expression of OPTN is altered in glaucoma patient. So altogether, this shows that OPTN malfunction may drive both anatomical and transcriptional changes that make this retina more vulnerable to degeneration. So now bring back to the focus, back to inflammatory cell death. So previous studies have shown that glaucoma patient carrying E50K mutation tends to develop a more severe and rapid progression form of disease. In addition, OPTN expression is reduced in a sporadic form of glaucoma cases. So OPTN malfunction may sensitize the cell to RIP1 dependent inflammatory cell death pathway. And as this mutation may alter the inhibitory protein of the RIP1 pathway. So to test this hypothesis, we move directly into the in vivo experiment using preclinical glaucoma model to evaluate how inhibiting RIP1 affect retinal inflammation and then neurodegeneration. So if RIP1 really is the central node, so we challenged the mice with two well-established preclinical models of glaucomatous damage. So the first is ischemia reperfusion injury. So this induced anterograde damage driven by metabolic and vascular stress. So we induced intraocular pressure to the anterior chamber of the eye, so it blocked blood supply completely. So, and upon restoring the normal pressure, reperfusion occur and triggering oxidative stress, mitochondrial dysfunction, and ultimately leading to degeneration of the RGC. So this model mimics the metabolic and oxidative stress components of glaucoma theory. In this model, RGC loss starts from the peripheral area of the retina. And the second is optic nerve crush, which mimic the retrograde damage resulting from the mechanical axonal insert. So to induce this damage, we access to the optic nerve of the mice intraorbitally without damaging the surrounding musculature, and then a fine forceps is used to clamp of the optic nerve approximately 1 millimeter posterior to the eyeball for about 10 seconds. So this induces retrograde degeneration of RGC and closely modeling the axonal degeneration seen in glaucomatous optic neuropathy. So in this model, the pattern of RGC loss is homogeneous, unlike IRI model represent. So the question is, what happens if we remove RIP kinase 1? If RIP1 is really the central decision point for inflammatory cell death, the next logical step is to test what happens when we disrupt its function. So to do this, we use the RIP1 kinase that mice that are genetically modified so that the RIP1 protein is present, but its kinase activity is deleted. And also by using this both model, we were able to test whether RIP1 inhibition is broadly protective across mechanistically distinct type of injury. So the question is, the RIP1 inhibition is protective in preclinical glaucoma model. So to answer this, we induce IRI and optic nerve crush in RIP1 kinase mice, allowing us to directly assess how inhibiting RIP1 affects retinal inflammation as well as neurodegeneration. So in this slide, how we examined how RIP1 inhibition affects the RGC loss in the IRI model. So we perform here immunostaining for BRN3 alpha and RBPMS as RGC marker. In wild-type mice, we observe a substantial loss of BRN alpha and RBPMS positive RGC cell in IRI model. However, in RIP kinase mice, these RGC were largely preserved after IRI damage. So we also examined SMI32 to visualize axons together with the cell body of RGC subtypes. In wild-type mice, 7 days post-injury, RGC cell body was shrinked or lost, or their axons appeared more thinner and degenerated. In contrast, RIP1 kinase mice retain the RGC cell body and shows a minimal axonal degeneration. And in addition, the OCT imaging confirmed that RIP1 kinase mice preserved the retinal thickness at 7 days of post-injury, while wild-type mice shows a significant retinal thinning. So this slide demonstrates that RIP1 inactivation provides robust neuroprotection against ischemia reperfusion stress in the retina. But how do we know if this survived RGC are actually still functional? So in order to examine if this functionality of the RGC is still intact, so we used two complementary approaches. One is a patent electroretinogram, so PERG, to measure the RGC neuronal activity, and the second one is optomotor response with optodrum from Striatech to assess the vision of the mice after IRI damage, and this visual signal transmitted to the brain. So here we can see representative PERC waveform in wild-type in rats and RIP1 kinase mice in blue across the condition. So after IRI damage, wild-type mice shows progressive drop in PERC amplitude, particularly day 7. But in RIP kinase-1 kinase mice, the signal remains intact. So the graph quantification of the amplitude shows that RIP1 kinase mice retain a significantly higher PERG response after injury, indicating RGC still firing. And next, we ask whether this functional preservation translates to the visual behavior response. So to do that, we use optodrum to examine the optomotor response, which measure the spatial frequency of mice response to the visual stimulation. And as expected, wild-type mice shows a dramatic decline in OMR measurement after IRI damage, while RIP kinase-1 kinase mice maintain their response near the baseline. So by combining PRG and OMR, we captured both cell intrinsic function and behavior vision outcome. So this dual assessment shows that RIP1 inactivation not only preserve RGC structure, but also maintain the visual function both at the electrophysiological and also behavior level. So PRG tells us the RGC are still active. OMR tells us the brain still sees, and structure without function is not meaningful. So this is a very valuable readout that we examine afterward. And also this data confirms that RIP1 kinase protects both structurally, functionally. So to test whether the protective effect of RIP1 inactivation generally beyond the ischemic reperfusion injury, we test a second model, optic nerve crush, which induces mechanical retrograde RGC degeneration. So here we show retina plate mount like previous slide, and in wild type mice we observed substantial loss of BRN3 alpha and RBPMS positive neurons, which is RGC, as well as a reduction of SMI32 labeled axon and density. And SMI32 positive RGC cell body does not really show significant shrunken cell body morphology in optic nerve damage, in contrast to the IRI model in wild type. In RIP1 kinase-mediated mice at 7 days post optic nerve crush injury, mild reduction of RGC death, but the level of RGCs are higher than wild type after optic nerve crush injury. So, and unlike IRI damage, OCT measurement at day 7 shows no significant differences between wild type and RIP1 kinase-mediated mice. So while OCT did not really show major structure changes, RIP1 inhibition confer a mild neuroprotective effect against mechanically induced RGC death. Despite no change in OCT detected in retinal thickness, histological analysis revealed the significant preservation of both RGC somas and axons. So as we did in the IRI model, next we ask whether the RGC survived in RIP1 kinase-mediated mice after optic nerve crush are also functionally active. So to address this, we again use a PERG and OMR as a functional readout. So PERG amplitude shows that significant reduction in a wild type after the optic nerve crush, in contrast to the RIP1 kinase-mediated mice really retain the stronger signal, suggesting preserved RGC function. And using the optomotor response test, wild type mice shows a significant drop in OMR performance after optic nerve crush. However, RIP1 kinase-mediated mice maintain the OMR score that were close to the control level, including the untreated contralateral eyes. So together, the data shows that RIP1 inactivation mitigate optic nerve crush induced injury that shows a visual dysfunction, and also the RGC remain functionally responsive and behaviorally relevant even after mechanical injury. So interesting part here is, although optic nerve crush reduced RGC numbers about 35% and 40% compared to the control eye, so RIP1 kinase-mediated mice still retain enough functional RGC to support the intact visual performance. So this suggests that preserving just around 50 of RGC may be sufficient to maintain the vision for glaucoma patient, which is a meaningful implication for long-term visual outcome. So next, we evaluate whether RIP1 inactivation protects neuronal integrity, both ischemia reperfusion and then mechanical injury, which is optic nerve crush model. So as we wanted to understand how it modulates the inflammatory response at the site of the damage, meaning that specifically the RGC layer, so we performed the flat mount staining of the RGC using μN here in green as a neuronal marker. And after 7 days of the IRI and optic nerves model, and we test and visualize how RGC numbers altered, as well as at the same time how microglia infiltrated in a inner part of the retina. So in wild type mice, both IRI and optic nerve crush model shows a strong infiltration of EBA1 positive microglia into the RGC layer, which is a sign of injury-induced inflammation. In contrast to wild type in RIP1 kinase-STAT mice, this microglia response is significantly attenuated. So you can see fewer and less activated EBA1 positive cells in the RGC layer, indicating the suppression of the local inflammatory reaction. Also, in quantification confirmed that microglia counts in the RGC layer are significantly lower in RIP1 kinase-STAT mice compared to wild type across both model. So this result suggests that RIP1 kinase activity is a key upstream regulator of microglia activation and infiltration. And its inhibition directly reduces inflammation where it matters the most, which is the RGC layer in the retina. So far we have learned that RIP1 inactivation provides a strong neuroprotection across a different injury model, both functionally and also structurally. But the question still remained, at which step within this RIP1 pathway, especially in necroptosis, does this protective effect occur? So how about the downstream regulators of RIP1 dependent cell death pathways? So RIP1 plays a critical role in balancing TNF-mediated inflammation and cell death signaling. So if RIP1 is really the switch, how about RIP3 and MLKL? They are executioner, so as a downstream regulator for necroptosis. So are they really necessary for damage, or can we intervene the downstream of the RIP1? That is the next question. So our goal was to determine whether the neuroprotective effect of RIP1 inactivation is mediated specifically through the necroptotic pathway by further activating RIP3 and MLKL as a downstream regulator. Therefore, we designed the additional genetic in vivo experiment to directly target these downstream effectors. So now we wanted to determine where this protective effect of RIP1 is mediated through the necroptosis pathway. So specifically via RIP3 and MLKL, which is the key downstream effectors. So to address this, we use a two independent knockout mouse line, which is RIP3 knockout and MLKL knockout mice, and subjected them to the ischemia reperfusion injury in the data is in this slide. So images panel A to C shows retinal flat mount stained for RGC in green and microglia in red. So wild type animals shows significant RGC loss after IRI, while RIP3 knockout mice retain a substantial number of RGC. MLKL knockout mice also show some preservation effect, though the less suppression of microglia infiltration and higher neuronal loss than RIP3 knockout mice. And PERG traces and quantification in panel D and E shows only partial functional rescues in RIP3 knockout mice, but not in MLKL knockout mice. So in panel F, again, OMR measurements result shows a similar trend. RIP3 knockout mice retain significantly better visual responses than MLKL knockout mice. So together this data, these results indicate that RIP1 contribute to the ischemia reperfusion mediated retinal damage, but it is not really exclusive node for RIP1, as we see higher protective effect from RIP1 kinase that mice after IRI. At the same time, MLKL may not be the main effector, including damage in this model. So RIP3 knockout protects RGC against RIP ischemia reperfusion stress, and structurally, also functionally. But MLKL knockout retained the number of RGC with less microglial infiltration, but visual function was just partially protective. So this approach suggests really MLKL independent role for RIP1 and RIP3 in this model. Then how about optic nerve crush model? We tested RIP3 knockout and MLKL knockout mice in this optic nerve crush. So both the knockout lines exhibit the neuroprotective effect by showing the higher number of new and positive RGC in compared to wild type following optic nerve crush injury. But only RIP3 knockout mice shows reduced microglial infiltration compared to wild type. Quantification in panel G to H support this observation, indicating reduced inflammation and also neuronal loss in RIP3 knockout mice under optic nerve crush injury. However, when we examine RGC function by PRG and visual behavior response by optodrome, it shows in panel I to K. So the PERG amplitude and OMR level were really compromised as much as wild type following optic nerve crush. Both RIP3 knockout and MLKL knockout mice did not really show functional protection effect. So MLKL knockout again shows no substantial protection functionally, but still number of RGC cells was higher than wild type mice after the injury. So this data also tells us that in the optic nerve model, RIP3 deletion helps retain the number of neurons, but it is not really sufficient to preserve a complete function. This reinforces the finding that RIP1-driven degeneration may involve additional mechanism beyond the classical RIP1, RIP3, MLKL necroptosis axis. And RIP3 knockout and MLKL knockout is not significantly protects in optic nerve crush model, while we observe the higher number of new and positive RGCs. And survived RGC does not really shows a good functional activity evaluated here by PERG and OMR. So here we tested RIP1 inhibition, really provide a neuronal cell protection. So, and also RIP1 knockout is protective in RGC in IRI, functionally and also structurally, yet it shows only modest effect in optic nerve crush. So MLKL knockout demonstrate a limited protective effect in both the models, suggesting the downstream effector of RIP1 are only partially responsive. So in contrast, what stands out across all our data is that RIP kinase inhibition provide the most robust and consistent neuroprotection, including preserving RGC structure, function, and visual behavior across both ischemic reperfusion and mechanical injury model, together with suppressed microglial infiltration. So given this strong protective phenotype, we then ask a more upstream question: what makes this RGC susceptible to RIP1 activation in the first place? So since OPTN mutation are genetically linked to the glaucoma and also reduce OPTN level in human glaucomatous retina are reported. So the next step was we explore how OPTN loss or the E50K mutation could modulate RIP1-driven cell death pathway. So the question is, what drives this vulnerability to RIP1-dependent cell death in glaucoma? Is necroptotic cell death enhanced by the OPTN absence or E50K mutation? So first we ask whether OPTN loss or glaucoma-associated E50K mutation affects cellular response to the TNF-alpha stimulation. So we use the CRISPR-generated OPTN knockout and E50K lines. So we combined two different combinations of small guide RNA to generate OPTN knockout lines, and immunoblot data confirming this efficient OPTN deletion. And these lines are all sensitive in necroptosis inducers. Afterward, and then we introduce OPTN wild type construct or OPTN E50K construct as a knock-in line. So then we stimulate this line with the TNF-alpha and monitor the inflammatory pathway activation. So as I illustrated before, the primary response of TNF-alpha stimulation is NF-kappa B activation. So in this panel A and B, we see that both knockout and E50K harboring cells shows enhanced phosphorylation of P65 and J and K upon TNF treatment. So this indicates that these cells respond more toward pro-inflammatory status in TNF-alpha stimulation. To further validate the pro-inflammatory phenotype, we performed the qPCR analysis of the TNF-alpha-induced cytokine gene expression. So panel C shows the result from the knockout line. Upon TNF-alpha treatment, we shows robust upregulation of the key inflammatory transcript, including interleukin 1B, CXCL1 and 2, TNF-alpha, interleukin 6, and CCL2. So similarly in panel D, we access 50K line. These cells also shows a higher induction of inflammatory markers. So in this slide taken together, this data support our hypothesis that OPTN dysregulation, whether by deletion or mutation, sensitizes the cell to TNF-alpha driven inflammatory response, likely through the dysregulation of RIP1 associated pathways. Then the next question, then how about RIP1 dependent cell death pathway? How this OPTN variant affect to that path? So next we ask whether OPTN and E50K mutation enhance RIP1 dependent necroptosis. So we treat necroptosis inducer called TBE, here consisted of TNF-alpha, BV6, which is inhibitor of cellular apoptosis protein, and mnicosan, which is pan caspase inhibitor, or TTAE together with takinix, which is TBK inhibitor. So also we use here RIP1 inhibitor GNE684 and RIP3 inhibitor GSK872 to confirm this induced pathway are really RIP1 and RIP3 dependent. So first we check the cell death signaling in the knockout lines. So we observe a significantly higher level of LDH release across both necroptosis inducer, TBE and TTAE. So this elevated LDH signal was suppressed by RIP1 inhibitor as well as RIP3 inhibitor. So we confirm here that knockout, OPTN knockout, enhanced RIP1 dependent necroptosis, and these are RIP1, RIP3 axis dependent. It's confirmed by those inhibitor treated condition. Then the next question, then how about molecular signaling when there is no OPTN? So immunoblot analysis confirmed increased phosphorylation of RIP1, RIP3, and MLKL, showing the strong activation of the necroptic cascade in the absence of OPTN. Then how about glaucoma causative mutation E50K? So OPTN E50K expressing cell shows a similar pattern than knockout line. So these cells also exhibit increased sensitivity to necroptosis inducer. And then those increased sensitivity also inhibited by RIP1 and RIP3 inhibitors. So immunoblot data also again confirm that higher phosphorylation of RIP1, RIP3, and MLKL, meaning E50K mutation enhance necroptotic cell death. So here we confirm that E50K mutation is a phenocopy of OPTN knockout line, as it responds in a similar way than knockout lines in cell death context. So the next step is to test if OPTN E50K carrying RDCs is more sensitive to IRI damage, and if we can block it with RIP1 inhibition. So as is shown in the left side, we visualize the SMI32 immunostaining as a marker for neurofilament in RGC cell and their axons. So the top row are control mice expressing a control payload, and the middle is expressing wild type construct, wild type optineurin, and bottom part is the retina expressing E50K mutation. Upon IRI injury, E50K expressing retina shows much more severe disruption in axonal morphology, as well as a cell body washerent compared to control, and also compared to OPTN wild type expressing retina. When we treated the RIP1 inhibitor together, axonal degeneration and cell death associated morphology were significantly attenuated. So let me summarize how different knockout mice for different necroptosis component affect neuroprotective effect across two preclinical glaucoma model. So we have this ischemia reperfusion model, which represent the metabolic stress via vasculature intervention. So RIP1 kinase death provide a robust protection across all three metrics, structurally, functionally, and in terms of inflammation measured by microglial infiltration. Especially RIP1 inhibition by GNE684 also attenuated axonal damage in E50K transduced mice, confirming its protective effects even in genetically sensitized model with E50K. RIP3 knockout shows a similar protection, but less than RIP1 kinase death mice shows. It suggests that the RIP1, RIP3 axis is critical under IRI damage. MLKL knockout provides only mild structure and functional protection with a partial suppression of microglia activation in IRI damage. So in contrast, in the optic nerve crush model, RIP3 knockout was structurally protective, but did not fully preserve RGC function and reduce inflammation. At the same time, MLKL knockout has no significant protective effect in optic nerve model. So in this slide highlight that RIP1 is a most effective intervention across both injury types, while RIP3 and MLKL play very context-specific role in glaucoma animal models. So as a last slide, let me summarize what I have shown in this study and this slide in this presentation. So we demonstrate that RIP1 play a central role in RGC degeneration under glaucoma animal models. So using these two distinct preclinical models, ischemia reperfusion injury and optic nerve crush, we constantly observed that RIP1 inhibition provides neuroprotection, both structurally and also functionally. So Importantly, RIP3 or MLKL knockout offered only partial or minimal protection. It also suggests that RIP1 acts through additional non-classical necroptotic pathway and inflammatory mechanism. And also we further validated this in a genetically sensitized context using E50K transduced mice, where RIP1 inhibition reduced the axonal integrity, increase the protection effect for the axonal integrity. This also supporting its therapeutic relevance. So finally, we shows that OPTN malfunction, either via knockout or E50K mutation, it really enhances RIP1 dependent inflammatory signaling. It confirms that RIP1 is not only a downstream effector, but also a key junction integrating genetic risk as well as a stress response in glaucoma. So in the end, the message is clear. RIP1 is a druggable molecule that integrates inflammation, necroptosis, and also genetical susceptibility in glaucoma. So targeting RIP1 offers a broader upstream therapeutic strategy beyond intraocular pressure control, and especially for progressive or normal tension glaucoma, where the inflammation and neurodegeneration really dominates. So our approach using two preclinical models that incorporate the two different types of stress, it demonstrates the potential translational relevance of RIP1 inhibition. And it also positions RIP1 as a strong therapeutic target at the intersection between inflammation, degeneration, and also genetical vulnerability in glaucoma. I'd like to close this presentation with expressing my appreciation for all co-authors. And finally, thank you all for your attention and your interest in this work. Thank you very much.
Thomas, you're muted.
Does it work now?
Yeah.
Okay. All right, I'm sorry. So I thanked you for your talk, and I mentioned that we already had a couple of questions coming in, and I want to encourage the audience to ask even more. So I want to dive right in. So the first question was why you used two different functional readouts, the pattern ERG and the optogram.
So we really want to examine the functional aspect of RGC. So that's why we use both readout to address a different aspect of vision when it's functioning. So PERG captures the RGC firing, meaning the RGC intrinsic electrical activity at the retinal level. At the same time, optomotor response really evaluate visual behavior, which is the brain actually perceive. So using both really allow us to validate not just the structure protection, but also functional preservation at both this cellular level as well as the system level. Mm-hmm. And also our model ischemia reflex fusion and optic nerve crush, how they work as an insult are quite different. So those both readout really complementary one from another, so that we have really comprehensive understanding this therapeutic approach, if it works or not, and eventually mice can see or cannot see.
Okay. So your optic nerve crush model, you would expect, I would assume you would expect ganglion cell death. So the question is, why did you not see a reduction of the thickness of the retinal thickness in the optic nerve crush model?
Yeah, so that is a really important point. So optic nerve crush analysis, it's this model. The insert is coming from back of the eye. So this OCT analysis that we adopted in this study primarily detects the change of whole retinal structure, whole retinal thickness. So in optic nerve crush, the degeneration is a more axonal injury driven from the mechanical damage. So it is a highly likely affect RGC layer primarily rather than affect the whole retinal area. So we might be able to see if we can analyze only very inner part of the OCT images, which is RGC layer, rather than analyzing the whole retinal thickness. But in this study, we focus more on anatomical change in the whole retina in general. So it's might miss some resolution that the ganglion cell layer alteration in OCT image. So I believe that this is why we do not see the huge change in OCT image analysis.
Mm-hmm. Okay, then there's a question that came relatively early. You might have answered that, but I'll ask you anyway. And that is whether the neuroprotective effect is due to reduced inflammation or a direct protection of the retinal ganglion cells.
So I think it's a really key point of the study. So while RIP1 inhibition clearly reduces inflammation, we also see the structure and functional protection of RGC. So it includes the preserved axonal integrity and visual function. So our data really suggests a dual mechanism. So RIP1 inhibition plays a role in both cell types. So RIP1 inhibition provides neuroprotection both by suppressing microglia-mediated inflammation and also by directly preventing RIP1 dependent cell death in RGC. So in short story, the protection effect of RIP1 inhibition acts in both cell type together.
Okay. Mm-hmm. And okay, and maybe that's then the same answer here. The next question was, could RIP1 inhibition affect non-neuronal cell types in your models?
Yes, because we think RIP1 expression is really expressed in many glial cells as well, including microglia, and especially RIP1 expression in microglia is particularly high compared to any other cell type. So we observed the reduced EBA1 positive microglia infiltration with RIP1 inhibition. So it's suggesting that this is a part of effect in mediating through the modulating glial behavior, not only microglia, but also myeloglia, and then many other surrounding cells. So it's not just a neuronal intrinsic protection. So yes, RIP1 inhibition expected to be affecting other cell type, and it's also highly interesting to study further in this regard.
Mm-hmm. So maybe we have time for maybe 2 or 3 more questions. So why do RIP3 or MLKL knockout show only limited protection? Why do you think that?
Yeah, this is an interesting point and important point as well. So based on our data, optic nerve crush causes retrograde axonal injury and mechanical stress, which may activate the cell death pathway beyond RIP1, RIP3, MLKL necroptosis. Because this is the mechanistic stress, basically you clamp the optic nerve. So compared to IRI, we expect that there are much more other cell death pathway play a role. But the IRI model responds more robust way to RIP3 and MLK deletion because it's ischemia reperfusion damage driven, which is also well known that RIP1 plays a role. So also this discrepancy suggests that RIP1 really mediates broader damage responses than RIP3 MLK in optic nerve crush. And also what's happening in optic nerve crush is likely more non-canonical or beyond the RIP1 pathway as cell death pathway.
Mm-hmm. So we had another question just coming in that I think is quite interesting. So if you think of using what you found in a therapeutic context, how would you go about in a human patient to actually inhibit RIP1?
Yes,
practically speaking.
It's a great question because I have thought a lot about it, how we can introduce RIP1 inhibition. So my thought is glaucoma is really age-related drug and it requires lifestyle medicine treatments. So I think if we can administrate orally and we can increase the constant level of RIP1 inhibition level by administrate the small molecule through oral administration and block this RGC that sustain RGC survival, at the same time suppress immune activation, that could be way to go. It means we need a small molecule that potentially can go through a blood-brain retinal barrier, a blood-brain barrier, and blood-retinal barrier together. So this is really something we have to keep in mind when we wanted to treat the patient, how we can administrate. So my short answer is going to be small molecule, administrated orally rather than injection, and sustain a constant level of RIP inhibition in plasma as well as eye in locally.
Mm-hmm. Okay. And maybe the last question before we have to wrap up. What cell types or cell type did you use to test the OPTN knockout and OPTN mutation?
So in this study, we use a 661W cell type, which is a murine retinal cell.
Mm-hmm. Okay. All right. So thank you again. Kyoung, I would ask you to stay in the meeting for a little bit afterwards, but to the audience, I want to thank you very much for attending. It was a great pleasure to have you here, and I hope you will join us again for our next journal club sometime later this year. Thank you very much. Bye-bye.